All right. So last time we were here, we talked about muscles. And I want to remind you all of how muscle cells worked. Like one of the big characteristics of our muscle cells was that they were both excitable as well as conductive. So that meant that if we hit them with a stimulus, that they would be able to conduct that stimulus down the length of their plasma membrane, right? And it did that, remember, through usage of voltage gated potassium and sodium channels. Okay. So that those channels would only open whenever it sensed that there was a change in voltage near it. Okay. And the change in voltage came from it receiving some sort of neurotransmitter like acetylcholine or something like that, which told some chemically gated channels to open. So acetylcholine hits chemically gated channel opens, changes the voltage in that area, which then causes the areas near those to change voltage. And then that signal that says, okay, now that this area has changed, now the next area changes, the next area changes, and so on and so on and so on down the length of the muscle cell. Well, hopefully that all sounds familiar, and hopefully you remember that fairly well, because that is literally how nerves work. Okay. So what we're going to do now is we're going to go into a bit of depth, a bit of detail on some nerves. And we're going to kind of start the same way, where we're going to basically talk about kind of functions and how we get to those functions and how we split things up and talk about it and all that kind of stuff. Okay. All right. Now, the nervous system, its first main goal is going to be its main goals are going to be to collect information, process that information, and then respond to that information. So we collect information through what we call receptors. Receptors will start this whole cascade of reactions whenever they respond to a particular stimulus. So each receptor is going to respond to a very particular stimulus, whether it's touch of a certain type or temperature or stretch, whatever it may be. That then causes that receptor to send a signal through that nerve or through a nerve, which will then get sent to either the spinal cord or the brain. Okay. Or both, really, the spinal cord and the brain. Okay. Now, the spinal cord and brain will then process and evaluate that information. Okay. So it will basically take in all of these sensory information that's coming in from all of your sensory receptors in your body at once. Okay. So that right now I understand that the thing that I'm leaning up against is actually fairly cold, that my body is actually fairly hot. My heart rate is relatively high since I just walked from Woodfin and then ran over here and ate literally as fast as I possibly could. I heated and ate my 11 or 12 ounces of food in the span of like 3.5 minutes. Okay. It was both quite hot, parts of it were also still cold, and I was eating it literally as fast as I possibly could, and then ran in here. Okay. Now, it is processing all of that kind of stuff. Okay. Whether it's temperature, touch, and then putting it all together. Now, your brain is going to shortcut a bunch of this stuff and not kind of have you be constantly aware of every single sensory receptor in your body at any one given time, because a lot of that information is unimportant in this particular moment. It is unimportant to me that I'm wearing a shirt and that my sensory receptors are feeling some amount of touch from the shirt that is currently sitting on my back and on my shoulders. That information is not important because that information has been the same since 5:45 this morning. Okay. However, when something changes, that then is when it will make you aware of that stimulus and start sending signals, as well as your brain will become more aware of the signals that it's receiving around those areas. Okay. But it's all up to the brain and the spinal cord to process and interpret and evaluate that information. Say, is some of this dangerous? Is some of this the type of information that we need to act on and act on now? All of those kinds of things. Okay. Now, we also have to respond to that information. So we then will have a different set of nerves. So some nerves that are going to go away from the central nervous system that are going to send nervous output to things like muscles to tell us to move, or glands to tell us to sweat or secrete saliva, whatever it may be. Those things, your glands, your muscles, those are all effectors. They're going to cause something to happen. Okay. Receptors receive or detect stimuli and send information to the central nervous system. Effectors are going to be things that are going to receive nervous stimulus and cause something to happen. Okay. Now, our nervous system, just like everything else, is going to be organized. We're going to organize it into the central nervous system and the peripheral nervous system. The central nervous system is your brain and your spinal cord. That is it. The brain and the spinal cord. The peripheral nervous system is your nerves and your ganglia. So if it is a nerve, it is part of the peripheral nervous system. If it is the brain or spinal cord, it is part of the central nervous system. If it is a nerve that extends from the brain or the spinal cord, it is still part of the peripheral nervous system, even if they're closely related, even if they're very, very close together in space. If it is a nerve, it is part of the peripheral nervous system. If it is not, it is part of the central nervous system. Okay. Now, we can also talk about them functionally. Functionally, we have two major groups, or I guess three major groups. We have the sensory portion of our nervous system, we have the motor portion, and then we're going to have an integration portion. Okay. The sensory portion is going to deal with sensory information. Okay. Whether it is sensory information coming from your body, so from various receptors from something like, say, your eye with all the visual information, your nose for smell, your skin for touch, your skin for things like temperature, your gut for, hey, I'm hungry. Okay, that's actually more visceral. I'm trying to think. Your proprioceptors, so the sensory receptors in your muscles and in your tendons telling you where your arms and legs are in space. These are all types of things that we are going to associate with the somatic sensory portion of the nervous system. The visceral sensory information is going to come from things like your organs. Much of this kind of stuff you're not really going to be aware of, okay? This is saying, hey, your blood sugar is low. Your CO2 concentration in your blood is high or low. That type of stuff. Stretch that's being placed on smooth organs and stuff like that, or smooth muscle organs. All of those kinds of things are going to be examples of visceral sensory information. So internal organs would be visceral. Your senses, your special senses and general senses are all going to be examples of somatic. Now, as far as motor stuff is concerned, this is going to be the portion of the nervous system that's going to cause something to happen, whether it's telling a muscle to move or telling your heart to slow down or speed up, telling a gland to secrete more of something or telling a gland to stop secreting something. All of those are going to be examples of autonomic, okay? So cardiac muscle, smooth muscle, glands, things that you cannot exert any sort of conscious control over, like I can't tell my heart rate to slow down, I have to do it a different way. That is an example of using my autonomic motor nervous system, okay, or my autonomic nervous system. Same thing with telling my stomach to squeeze and push the stuff out of my stomach into my intestines. That is autonomic. I can't exert conscious control over that. I can exert conscious control over my skeletal muscles, okay? So if you can exert conscious control over it, we are using the somatic portion of our motor nervous system. Now, another big similarity between muscles and nerves is the structure themselves, okay? So here we have a typical nerve, like so something that would extend out of the spinal cord. So this is a typical spinal nerve. So if you were to look at the brachial nerve or the axillary nerve or the femoral nerve, they're all going to look pretty similar to this, where you're going to see individual muscle cells, which are going to be covered in their own layers of connective tissue. Now, remember, or anyone remember what an individual muscle cell is coded in, what we call that connective tissue that covers an individual muscle cell? It is dense irregular connective tissue, but what's the name for it? Or dense regular connective tissue? So what's the name for it though? That connective tissue coating. It's on the inside of the muscle. So it is endomysium, where endo is that innermost layer, okay? So in this case, in our nerves, we have the single nerve axon with a coating of endomysium. Well, then we're going to have a bundle of those individual nerves and their individual connective tissues, and they're going to get bundled together and coated in another layer of connective tissue. This bundle, we're going to call a fascicle. Anyone remember what you call a bundle of muscle cells? What do we call a bundle of muscle cells? It's the same word. Nope, nope. It's coded in endomycin. Not the coding, just the name of the bundle. Mycium is the name for the connective tissue coatings. So a muscle fiber is a single muscle cell. A bundle of muscle fibers is called a fascicle. A bundle of nerve fibers, so a bundle of axons, is also going to be called a fascicle. Okay, so the bundle of individual fibers, in this case, will still be called a fascicle. And it's going to be coated in another layer of connective tissue. In this case, it's perimycium, just like it was in muscles. A muscle fascicle was coated in perimycium. A nerve fascicle is coated in perineurium. Okay, so NEUR instead of MYS. Okay, so endoneurium on the inside of the nerve, perineurium wrapping around a fascicle, and then we're going to have epineurium coating the outside of the nerve. The nerve is going to be composed of a bundle of those bundles. Okay, so we're going to take a bunch of those fascicles, we're going to bundle them together, coat it with a layer of connective tissue, and all together, we will call that our nerve. Now, there are some special spots in our nervous system where we're going to find these big bumps, okay? So what you're seeing up here bundled together in the nerve itself, this is simply one small part of the cell, of the nervous cell, and we're going to call that small part the axon. Okay, that's going to be the distance covering portion of the cell, so where we're sending the signal from point A to point B. That would be our axon. The cell body, so the main portion of the cell, is actually going to be found in either the gray matter of the spinal cord or in this big bump right here. We're going to call these big bumps ganglions. Okay, that's just because the cell body takes up so much space that we have to store them in very particular areas. So if it's stored outside of the central nervous system, so outside of the spinal cord, we're going to store it in this big bump right here that we're going to call a ganglia. Now, most of your spinal nerves, pretty sure all of your spinal nerves, are going to be what we call mixed nerves. These are going to be two-lane highways, or at least multi-lane highways that travel in both directions. Okay, so there will be information going from the central nervous system, so from the spinal cord out to the effector, and there will also be information in that same nerve bundle carrying information towards the central nervous system from some receptor. Okay, so if you damage one of these spinal nerves, you will shut off both nervous output and nervous input that's going through that nerve. Okay, there are some nerves, though, like those in our skull, so what we call cranial nerves. These are going to be some specialized nerves that are going to extend from the brain. These are typically going to be involved with our special senses or sensory information from our face and tongue and all that kind of stuff. But many of these nerves are going to be unidirectional, okay? So it will be carrying information just from the receptor toward the brain. So a good example of this is your optic nerve. So your optic nerve is going to extend from your eye directly back into your brain, okay? It carries sensory information. That's it. All of those nerve fibers will be carrying impulses from the receptors in your retina to the brain. No information is going to be going the other direction out of those. We don't have any effectors on the inside of our eyeball anyway, so that's fine. But, or at least not in that part. So all of the information that nerve is going to carry is going to be sensory. There will also be some nerves that carry only motor information. So once again, that's just simply going to be one-way traffic. However, none of those are going to be the spinal nerves. Now, your spinal nerves are going to be the nerves that are going to extend from your spinal cord out in between your vertebrae. So where your vertebrae interact with one another, we have that little space in between them that we call the intervertebral foramina. That is actually where your spinal cords are going, or your spinal nerves are going to extend from, or through is going to be going through that space in between the vertebrae. So where the vertebrae interact, there's that little hole in between, and that's actually where our spinal nerves are going to pass. Now, something that makes our neurons different from our muscle cells is that they are secretory. This means that they release a neurotransmitter. If we are conductive and we send a signal down the length of the axon, the way that we are going to get that signal then to go to the effector or to the next neuron in the chain is going to be through the release of a neurotransmitter. Okay, so we have to have this secretory portion. Okay, now if y'all remember, we had at our neuromuscular junction, we had this big bump portion of the neuron. We call this the synaptic knob. Okay. Now, in the synaptic knob, we're going to have some proteins that are voltage gated calcium channels. And there will also be some calcium pumps in here as well so that we can maintain the differences. But whenever the voltage change gets to right here, so we're going to go from positive on the outside to negative on the outside, or positive on the inside, whenever that happens, and we have it now more positive on the inside next to one of these, this thing opens, and when it does, it's going to allow calcium to enter the cell. Okay, so our calcium amount inside the cell is going to go up, and that's what's actually going to cause us to release these little vesicles, which will then release our acetylcholine or whatever neurotransmitter. Okay, we can't get the signal out of the nerve without having the voltage get to the end of it. Okay, but the way that we get the signal out of the nerve is through the release of these neurotransmitters, like acetylcholine. There are others. This is just one example. That's the one that tells muscles what to do, for instance. But we'll also tell nerves what to do and all that kind of stuff. Okay, so that only happens because we have conducted the signal to it, and then we release, so we secrete the neurotransmitter out of it. Okay. Now, something else that makes neurons interesting is that they live for a very, very long time. They will live your entire lifetime. Once they form, they will stay that way. They can grow. They can get longer as you grow taller and all those kinds of fun things. If you get hurt, they can repair themselves a little bit, but they don't repair themselves very well. They do not repair themselves as well as something like, say, your arm, where you scratch a portion of your arm and stuff like that, you get a little bit hurt. And see, I think I've got a scratch back here. What happens? Well, that scratch eventually goes away as the cells that were damaged just get replaced by new cells that we're constantly making from our basal layer of our epidermis, right? Eventually, we will make enough new cells that the damaged ones will all go away and we can no longer tell that there was a problem, right? That's not the case with neurons. These things live forever. Because they live forever just as a single cell, they do not have to divide. They are not replacing themselves constantly. Because they're not replacing themselves constantly, damage to these things can take a whole lot longer to heal. Okay. So most of the time, we are not making new neurons. We're just simply relying on the ones that we already have. That's also why it's so important for us to protect the neurons that we have. Now, when we look at a neuron, we are going to see this. So here is a structure of a typical neuron. This is a multipolar neuron. So we have lots of feelers. Okay. Now the biggest structure that you're going to see right here is the cell body. Okay. So we have the cell body. We have a nucleus. It's got the little round spot on the inside of the nucleus. That's our nucleolus. So that's where we make our ribosomes. But this region is going to be held in shape by a bunch of cytoskeleton filaments and all that kind of fun stuff. But also extending off of this cell body, you see a bunch of little feelers. These little feelers out here are going to be called dendrites. Okay. So these feelers are doing exactly that. They are reaching out into the environment or towards receptors or towards other nerves, looking for other nerves or looking for impulses. Okay. Looking for stimuli. Okay. So those regions, the receptive regions of the neuron, we're going to call the dendrites. Now, then our cell goes into this big, long, skinny line down here. This is called the axon, okay? The axon is this long, skinny portion that we're going to use to transmit a signal from up here. So if we start making a signal up here, that signal will get propagated all the way down the length of the cell until we get down here into what we call the axon terminals, okay? So where we split that axon into a bunch of, like, say, fingers, okay? Those fingers would be our axon terminals. Now, this part of the axon is actually not over-exaggerated in its size, okay? Or of the cell, it's not over-exaggerated in size. You will have axons in your body that are 4 feet long, okay? For instance, they will extend uninterrupted as one cell from your L2 vertebrae all the way down to the end of your little toe. And in my case, because I'm tall enough, that is the better part of 5 feet long as one single cell. Okay, that is insane. That is a long way to send a signal, considering we are using a whole bunch of little protein pumps to do this, down, like embedded in the cell membrane to send the signal. That is a long, long, long way to transmit a signal. That means we need a way to speed up that process. Because we're using those protein pumps, we may have 20 million or 100 million of those protein pumps between the start of the signal at the cell body and the end of the signal at the axon terminal. That's a long way. So how do we speed up that process? Because we don't want to we can't go from, we don't have time to go from one to the next, 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 to the next times 20 million. We don't have that kind of time. So instead, what we're going to do is the same thing that you would do if you wanted to speed up the transmission of a signal through a cable. That's that you cover it, okay? By covering the cables, you stop signal loss out and weird signals getting in. The same thing goes for these neurons. We're able to stop weird signals coming in or coming out of the axon. And in that process, we're also drastically speeding up how fast we can send or transmit the signal through it, okay? So the way we're going to do this is pretty simple. We're going to cover it with these little cells here that we're going to call the myelin sheath. These cells, if this is in a peripheral nerve, we're going to call these neurolymnocytes. If it's in the central nervous system, they'd be called oligodendrocytes, and they would look a little different. But we're going to cover the axon, which is that yellow portion, with this thin layer of what we call myelin. Now, myelin is basically going to be a bunch of cell membrane, which is composed of fats. Remember that our cell membrane is our phospholipids, right? Where we have more lipid than we do phosphopart. So because we have these phospholipids, and we line them up and stack them 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 like 20 layers deep, that whole region is basically fat, okay? Now, what that's going to do though, say we have a whole bunch of protein pumps right here, and this is what we're using to change or transmit the action potential from one to the next, okay? And allow us to change it from being positive on the outside to being positive on the inside whenever we start that signal. Well, when that happens, this next one would have to go, and then this next one would have to go, and so on and so on and so on, and change that signal at each and every voltage-gated pump. Through 5 feet, that's going to take forever. So instead, what we do is we cover big sections of it with myelin. Okay, now that signal needs to basically hit this one, this little protein pump right before the myelin sheath, and then it will jump to the next space between the myelin sheaths. So now we're able to speed up the rate that we can send signals down our nerves by 100, 1,000, 10,000 fold, just simply by covering it with this myelin. This also means, though, that say you have Guillain-Barré syndrome or you have some other disease that affects your ability to form or maintain your myelin sheath. When that happens, that means you cannot send nerve signals or receive nerve signals as fast, which can be very, very problematic because we rely on. Now, after the axon, we split, like I said, into these little finger-like projections. We're going to call these finger-like projections the axon terminals, okay? Those axon terminals, so this would be the axon terminal. And this would be the synaptic knob. So the finger portion would be the axon terminal. The actual end of the finger, the end of ET's fingers, are going to be the synaptic Okay, so those are the portions of a typical neuron. Now, we do have different types of neurons. We have multipolar neurons. That's kind of the drawing that I showed you a second ago. This is a cell that has one axon, but a whole bunch of dendrites. By far the most common type. Now, we do have some specialized neurons that will only have one dendrite, and then they'll only have one axon. These will typically be connected to a single receptor, a sensory receptor. We're going to find these in the eye, particularly in the retina of the eye. So you're going to have a photoreceptive cell that is connected directly to one of these bipolar neurons. Because, though, we only have one set of input that is going through this neuron, this also means that the signal from each and every one of these bipolar neurons can be traced back exactly to where the impulse came from. By having many bipolar neurons, your brain is able to be very particular on where each and every impulse from those neurons came from, which means we can build an incredibly detailed map of all of those stimuli. How detailed? Better than 10K on your TV screens. This is how we see. The reason that we can see in really high fidelity is because we have a whole bunch of individual neurons taking the information from one receptor straight into the brain. Your brain knows exactly where that information came from, so it can build incredibly detailed maps of that information that's coming in, of the light that's coming in, okay? That is very different from something like, say, in your skin. I can touch right here, and I'm probably touching 200 sensory receptors that are all sending their signal that says I'm being touched, and it's going to be localized to roughly here, okay? But what you'll find is that these areas are going to be much less sensitive to that information than something like your eye, which can put it down to like little tiny amounts of space between them, okay? That's going to be very different than something like in your skin, where it's not going to be nearly as distinct where that information is coming from. Now, your hands will still have multipolar neurons from their sensory receptors, but those multipolar neurons will be responding to fewer receptors than in something like your arm, which means we can map information for touch in our finger much more clearly than we can in something like, say, our arm or on our back. Okay, I just simply know that my shirt is touching my shoulders, but I couldn't pinpoint the exact hair that is being touched by my shirt because I just don't have that amount of sensitivity. And that's because, hey, we have a bunch of hairs that are all being innervated by the same set of neurons, okay? Now, we're also going to find some weird neurons in here as well. So these are going to be either unipolar or anaxonic. Anaxonic means it does not have an axon. So this is a neuron that's basically just a bunch of dendrites, just cell body, bunch of dendrites, just reaching out all around it. These, though, are going to be very useful in places like your central nervous system where we can make decisions and stuff like that. We also have a bunch of unipolar neurons where we have a single process coming from the cell body that will then split. So some big differences there. I'm not worried about necessarily your ability to identify all those, but I wanted you to kind of think or realize that not all of our neurons look like the drawing that was up there. Now, we do have some functional classifications of our neurons. These are important. However, we've already talked about it a little bit. We have a neuron that is carrying information from a sensory receptor, and it is going toward the central nervous system. So if we are carrying information to the central nervous system, we are going to be called a sensory neuron, okay? So these will take information from a receptor and send that information to the central nervous system. If instead it is carrying information away from the central nervous system, so telling a muscle what to do, telling a gland what to do, all of those kinds of things, those are all examples of motor neurons. Information away from the spinal cord goes through a motor neuron. Towards is sensory, away is motor. Now, in between those, we have the interneurons. Remember that inter means between, and interstate is called that because it goes between states, okay? So the interneurons are going to be the neurons that are in between the sensory neurons and the motor neurons. These interneurons are also the ones that are going to make decisions and interpret information. They will receive information from not just one sensory neuron, but lots and lots and lots and lots of other sensory neurons so that they can figure out what is going on in their particular little field that they are connected to. Okay. Now, say you touch something that is hot, like say you put your hand in a fire. Okay. Like you're in micro and you accidentally put your hand in your Bunsen burner. Don't do that. Those are not just a little bit hot. Those are incredibly hot. When you do that, though, that's going to be heat and pain information coming from your sensory neurons. They're going to go up here. They're going to interface with this interneuron. That interneuron is going to be like, hey, dummy, move your hand. And then it will send a signal immediately out through the motor neuron telling your muscles to move your hand out of the fire. The same time that it sends that signal towards your muscles, it will also be sending a signal up to your brain that says, hey, we are feeling pain and temperature at these particular receptors. But you'll notice if that's ever happened to you, that you will have your hand out of the fire before you have registered that your hand is hot or that your hand got poked by something. Your hand is actually already moved away from the stimulus. That is a reflex, okay? It is a reflex because that motion, you pulling your hand away, occurred because the interneuron just told the muscles what to do before the brain even got the information. The brain was bypassed. Hey, this is important enough that we can just move your hand and we'll let the brain know that that's what happened. Okay, so your brain will say, oh, hey, my hand got hot, usually well after you've already removed your hand from the painful stimulus. In the case, that is, like I said, a reflex. Now, importantly, our nervous cells are really, really good at sending signals, but kind of garbage at everything else, okay? They do their jobs really, really well, but in order to help them survive and actually help them do their jobs, they have a bunch of helper cells. These helper cells are going to be called glial cells. Now, the central nervous system is incredibly dense and thus is going to have lots and lots of specialized glial cells to help it do its job. Now, the first one of these is that we'll talk about is going to be the oligodendrocytes. We've already talked about the importance of making our myelin sheath. So we're going to make or form the myelin sheath. So it's usually going to be drawn in white. We're going to make that myelin sheath using these cells here, which are oligodendrocytes. But notice that this cell is myelinating this axon once, twice, and then it's also insulating this axon twice. So it can insulate multiple portions of multiple axons all at the same time, okay? This gives us some structural help because now we basically have these two neurons tied together in space, tied together and held there by this oligodendrocyte. So we have some structural help here, but the main thing that we're doing is myelinating multiple regions of multiple axons. That makes oligodendrocytes kind of interesting and kind of different than the ones that are doing this job in the peripheral nervous system. Now, we also have these little guys right here. By little, I mean gigantic. This is an astrocyte. It's called that because it looks kind of like a star. Notice that it has a bunch of feet kind of poking out all in multiple directions, kind of like a star. So this is our astrocyte. Now, the astrocyte has a bunch of little feet that will allow it to go out and touch multiple nerves in multiple places, but it will also extend special little feet out to touch the blood vessels in the nerves. By touching the blood vessels in the nerves, or in the central nervous system, it's going to wrap around those blood vessels with these special little fat feet that it will use to control the things that are going into or out of these blood vessels. It is forming what we know as the blood-brain barrier. Very few things can cross the blood-brain barrier. This is very important. It's important because we want the chemical environment inside the central nervous system to be very, very distinct from the other environments. Why? Well, because we are using the movement of sodium, potassium, and calcium to regulate all activity inside the brain. If those ion concentrations get messed up for any reason, because say too much salt floods into our brain, too much potassium floods into our brain, the nerves don't function. And if the nerves in your brain don't function, you die, and you die now. Okay? Or catastrophic things occur that you can never recover from. If you've ever known someone that's had a stroke, this is basically because blood supply got shut off to one tiny portion of their brain. or usually a small portion. If it's a big portion, they just die. But it's usually a small portion. Blood supply gets shut down to that one small portion of their brain, and those cells are without oxygen for a few moments, and you know it for the rest of their life. That is something that they usually never fully recover from. They can get close sometimes if it wasn't a particularly bad one, but it is still something that you can tell that this person has had a stroke for the rest of their life. All because of a little bit of blood supply in one spot got messed up. We maintain that barrier to help us maintain a very particular chemical environment inside the brain. We help maintain that particular chemical environment using these astrocytes, okay? Now, because these astrocytes are tying together things like cells and blood vessels and holding other cells in place with other cells, these are also very important for helping us maintain kind of the structural integrity of the brain. So the astrocytes are very important. Now, one of the other ways that we maintain the special chemical environment inside the brain is we don't have blood as our primary interstitial fluid in the brain, okay? We don't have these cells just floating in blood. Instead, these cells are suspended in what we call cerebrospinal fluid, okay? So cerebrospinal fluid is going to be the primary fluid that envelops all of the central nervous system. And that stuff is made by these little column-shaped cells right here. So these column-shaped cells that have these little feelers, these column-shaped cells that line all of the cavities in the brain, we're going to call these ependymal cells, E-P-N-D-Y-M-A-L, ependymal cells, okay? Like I said, these line all of the empty spaces or open spaces in the brain and spinal cord, okay? And they are also going to secrete cerebral spinal fluid into those places, once again, so that we have a nice, static sort of chemical environment in which our central nervous system lives and can kind of do its job, okay? So they're always going to line these spaces. Now, the spaces in the brain are going to be called ventricles. So if you ever see the word ventricle, it's basically talking about a space inside the brain. Now, lastly, in case something does come out of this blood vessel that doesn't need to be there, so say we do have some sort of pathogen or we have some cellular debris or something like that in the brain that needs to be taken care of, we have these special little immune cells called microglial cells that are going to help take care of this, okay? These are going to be cells that are going to act as phagocytes in our central nervous system. So cellular debris, potential pathogens, bacteria, viruses, whatever it might be, these are the things that are going to help get rid of those. Now, the peripheral nervous system is a little bit more straightforward. We are sending information to and from the spinal cord. That's it. We're not really interpreting any of that information, so we don't have to have billions of connection points with interneurons and stuff like that where we're decision making or anything. So instead, we have two main cell types. We have our insulating cells. In this case, these are our neurolymphcytes. You'll also sometimes see these called Schwann cells, but your book doesn't like using those kinds of names. It prefers to use functional names. So you're going to see these called neurolymphcytes. So neurolymphcytes are forming our myelin sheath in our peripheral nervous system. So you have one here, one here, one here, one here, and so on and so on and so on. And then up here in the ganglia, the bumps, so remember that the bumps are ganglia, that's where we have our cell bodies. Surrounding the cell bodies and help maintain the environments around those, we have these satellite cells. So a satellite, remember, is something that kind of orbits around something. So the same thing is going to go for these satellite cells. They're basically just going to orbit around the cell bodies in the peripheral nervous system and make sure that they are safe, regulate nutrients, waste, and all that kind of stuff for those cells. So satellite cells around the cell bodies and the ganglia of the peripheral nervous system, and then neurolymphcytes forming the myelin sheath. Now, myelination happens because we basically take a little bit of cell membrane and we start to wrap and fold that cell over the cell over and over and over and over and over again. Okay, so basically just continues wrapping around it and wrapping around it and wrapping around it to where we essentially have no cytoplasm anymore, where it's basically just cell membrane touching cell membrane layer, touching cell membrane layer, touching cell membrane layer, touching cell membrane layer. Okay, but remember that cell membranes are mostly composed of lipids. That's why these are always drawn in white. Okay, what color is fat in a steak or on chicken or anything like that? When it's cold, it's white. That's why it's drawn that way. It is drawn white on all these pictures because it is composed of fat. If you go back there and you look at that human brain, or next time we come in and we cut open a sheep brain, you're going to see lots of white matter and gray matter. The white matter appears white because of the lipids in the myelin. It is white matter because it is composed of myelinated axons, not cell bodies, but myelinated axons. Regions that are made of white matter are basically information highways. Information is going through it, okay? Decisions get made in places where you find gray matter, okay? So once again, central nervous system are oligodendrocytes myelinate multiple axons all at the same time. Now, I mentioned Guillain-Barré. This can be something that goes away over time, so that you can recover function. However, if it does not go away, you basically will lose access to muscle strength and all that kind of fun stuff. Multiple sclerosis is a much worse form. Basically, it means these oligodendrocytes get attacked by your immune cells, so you don't have oligodendrocytes anymore, which means you don't have myelination in your central nervous system, which means we vastly slow down the rate at which we're able to send nervous signals through the brain, okay? Which can really inhibit our ability to do any sort of peripheral motor function just simply because we can't get those signals out in the right orders and stuff like that. So things can get a little bit wonky for those people. They tend to think okay, but as far as moving and stuff like that, you tend to run into some issues. Now, if an axon does get hurt, it can get repaired. So I do recommend that you read up on figure 1210. This is basically just showing you kind of the repair process for axons that do get damaged. However, it does usually, or this process can take a very, very long time. And in the central nervous system, it may not occur at all. And we've just filled those spaces with astrocytes. Now, once again, we are relying on voltage-gated channels in the axons and axon terminals, voltage-gated channels in the synaptic knobs to allow us to open up and to continue propagating the signal down the length of the cell. We're going to find those voltage-gated channels in the regions that we are trying to transmit signals through very, very quickly. Throughout the entire cell membrane, though, we're going to have some special things. We're going to have sodium-potassium pumps, which are going to try and maintain our resting membrane potential, basically maintaining this idea that we are more positive outside the cell than we are inside the cell. By maintaining that, or help us maintain that, we have to have a pump. This means that we're going to be pushing things against their concentration gradients so that we can be more positive outside than inside, because if you just let everything just go to equilibrium, it would be equal. So to maintain that difference, we're going to be using these sodium-potassium pumps. So you're going to pump 3 sodiums out and 2 potassiums in, making the outside more positive than the inside, okay? However, also helping us maintain these things, because we will overshoot quite often, we will have some leak channels that will allow things like sodium and potassium to move from the outside to the inside or inside to outside based off of what those concentrations are. This way we can maintain that resting membrane potential so that we can maintain our ability to be excited. If you ever get to the point where the membrane potential is swapped or is not held at its resting rate, it cannot be incited to send a signal down the length of it. We have to have this difference at the start, otherwise we can't send the signal through the nerve. So we spend lots and lots and lots of energy daily on maintaining the membrane differences in our nerve cells and in our muscle cells. Okay. Now, the sodium-potassium pumps and those leak channels are going to be found through the entire length of the entire axon. The voltage-gated channels, we're going to find in what we call the conductive segment. So going from our axon hillock all the way down through the axon, that is where we're going to find these voltage-gated channels. Because if we change voltage here, we'll change voltage here, and here, and here, and here, and here, and here, and here. And that's how we send the signal down the length of the nerve, okay? However, up here, where we are making the signal in the first place, in the dendrites and in the cell body, up here, we are not voltage gated. Instead, up here, we are chemically gated. That's what those little words are that you can't read because they're tiny. Up here, our proteins are chemically gated. They will only open and cause a change in the membrane potential whenever they are hit with a particular chemical, which comes from the synaptic knob of another nerve. Okay. Or from a conformation change in the receptive portion of their dendrites or something like that. Okay. So another nerve can talk to this one, and if it secretes its acetylcholine, its neurotransmitter, that then will cause these chemically gated channels to open, and then us to change our membrane charge right here, change our membrane potential at this spot. And if we do that enough, that we cross some sort of threshold barrier here in our axon hillock, then all of a sudden we will open up all of these voltage gated channels, and they'll just open. Once we hit the voltage threshold of the voltage gated channels here in our axon hillock, we send the signal. Once it starts, we can't stop it. It's all or nothing. Just like when you pull the trigger of a gun, it doesn't matter how hard you pull the trigger, it goes off. Once you cross the threshold of how much pressure you apply, it fires. The same thing goes for this. Once you cross the threshold, once you cross that sort of initial threshold for these voltage gated channels, it will go. Sends the signal down the length of the nerve, and you can't stop it once it starts. Okay, but you have to reach that particular threshold. That is how nerves work. Once we get down here to the synaptic knobs, instead of it being voltage gated sodium potassium channels, instead we have those voltage gated calcium channels, and that's when we're going to start releasing our secretory vesicles because of calcium. So once again, we have calcium playing a vital role. Okay, now the reason I say that this is how how nerves work is because in your heart, we have some specialized muscle cells that will do this same thing. However, instead of it being where we have our resting membrane potential and then our threshold value, where we're held here and then we receive a signal. Actually, this resting threshold value would be like here. And then we receive a signal and it's like, oh, we go, oh, and then we fire. And this is how nerves work, where there's very little change here before we set off the thing. We have some specialized cardiac muscle cells that will be very leaky, okay? Where those leak channels are open a lot more often, and they're just open all the time. So whenever you have had a firing event, it'll get back to its resting rate, and then it'll just slowly creep back up, hit a threshold, and then go. Creep back up, hit a threshold, go. Slowly creep back up, hit a threshold, go. And it's going to do this over and over again. This is how your heart sets its own rate. Your heart, if you give it no nervous innervation at all, will beat around 70 beats a minute. Maybe even 90 beats a minute. It requires nervous innervation to slow it down. Okay? Because it just wants to beat at that particular rate. It does that because of some leaky channels found through the entire length of the cell that will slowly allow it to creep towards that threshold value. And once it hits the threshold value, it sends the conductive signal down the rest of the length of the cell. And then it tells, well, all of the other muscles that are attached to it, your cardiac muscles, tells those to contract, which means our heart beats. Okay. So by learning about it now in the neurons, it will help you a bit. There's some stuff here about Ohm's law and resistance and stuff like that and nerves. So that's something you might want to pay attention to. But that's more physiology than anatomy. I'm trying to get to anatomy stuff. All right. The last bit of anatomy that I want you all to realize is that we have lots of nerves talking to each and every nerve, especially in our central nervous system, all at any one given point in time. Each of these little ET fingers you see right here is a different nerve talking to this one, potentially telling it to fire or not. We will have some of these which are excitatory, some of these which are inhibitory. The excitatory ones are sending signals through them or can send a signal through them that says, hey, you should go off. The inhibitory ones are sending signals that said, hey, you should not. And these two signals are fighting constantly, okay? Depending on what the input is from these nerves. Hold on. Depending on what the input is from these nerves. If we get more additive impulses from the excitatory ones, and those numbers add up to where we get enough charge to where we get above our threshold potential, nerve fires. That's it. It's all additive. Okay. If you think of the green ones as positive and the red ones as negative, if you end up with more positive signal than negative, it will fire. If you end up with more negative signal than positive, it won't. Okay, so we're constantly having this battle of should the nerve send a signal or not, based off of the information that's coming to it. Okay. All right. What was your question?
Excitement in the body that causes that. So that where the nerves are constantly firing.
So you do reach a point where it's like anytime you're trying to send, like, say we're trying to tell a muscle to contract. We're going to be sending impulse, 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 where each and every one of those impulses is telling the muscle to maintain some tension or to fire and contract. Your muscle will only relax whenever we stop sending the signal through it. However, we can only do that so fast. You do have a period in between which that your muscle or that the nerve fiber cannot send another signal. So sometimes you do reach these points where it simply can't get another signal going out. And part of that is because we're relying on these charges. Like say we received another stimulus right here. Well, if we receive another stimulus right here and we're already depolarized, we're not going to be able to depolarize again because we're already depolarized. We have to wait until we're back here to our resting rate before we can do it again. So there is a little bit of like, we can't continuously stack the signals on top of one another. We have to wait until this one is over before we can send another signal. However, as far as the muscle cell is concerned, it's constantly receiving the signal that it needs to stay contracted. So it will continue receiving the signal to stay contracted until the nerve signal stops. Once the nerve signal stops, that's when the muscle moves. But yes, there might be times to where it's getting signals from a lot of these. But if it's getting a whole bunch of inhibitory signal and a whole bunch of excitatory signal, it's truly just going to be additive. It's whichever number wins what's going to decide if it does it or not. Now, that might be different than what you're talking about, where it's like, hey, I cut off my leg. That's a ton of pain that's coming through there. Eventually, it's so much pain that your brain can't handle it and you pass out, but that's a different sort of thing. Did I approach the question that you were asking, sort of?
It's more related towards muscle and understand that. That's why we get cramps. In some way.
Sort of,
yeah.
Not really, but
depending on what that.
Mhm.
I was just thinking in general, the body with nerves sending out impulses like itching response.
Mhm. Yeah, a bunch of that is truly just like that's just all signal dependent. And sometimes, like when we get to senses, we will talk about neurons or sensory receptors that stop sending out impulses whenever there's not a change in signal. We'll talk about those sorts of things Monday. So we will get there. But for now, I just want y'all to understand that a single neuron is going to be constantly trying to tabulate basically all of the information coming in from all of its various inputs before it makes a decision. That this process is additive or summative. Okay, so basically adds it all up. If we cross the threshold value, then it goes. If we don't cross the threshold value, which is around -55, then it won't. So if we get to -52, doesn't fire. Or actually, hold on, -52 is above that, so it would fire. If we get to -60 or -58, it won't fire. Okay, it has to reach this -55 before we'll go. All right. Okay, so I think that's it on the stuff that I wanted to talk about today. So there are a couple of virtual labs for chapter 12 where they will go into this process and you can kind of see the connection between this and this at particular spots along the axons and kind of understanding how all that stuff is going to work. So make sure we do those. There's really not a whole lot lab stuff that we can do. Like we could look at a nerve fiber under the microscope if you want, you can. There's not a whole lot to see. The cartoons are a little bit better for seeing the types of structures that I want you all to see rather than the actual microscopes will, just because those tissues are busy, to say the least. Okay, everybody good? All right, then we're good to go. When we come back next time, we get to cut open a brain. See you Wednesday.
So as the body wears out, how does the myelin, does it stay intact?
It should.
Good.
Yeah, it should stay intact. Yeah, because it doesn't, like, these cells are made to live basically your entire life, and once that stuff is set up, it should be there. Now, if the myelin sheath gets damaged for whatever reason, it does take some time to repair it, but it usually can be repaired.
Just curious. I enjoyed it, and so I appreciate you answering my question. You'll have a good day.
Oh, you too.
Just wondering, when was the, were you gonna grade the module 2 exam?
Yes, it's being graded now. I'm just finalizing the last few questions, and once I'm done finishing those, they'll get posted.
Okay. All right, well, I'll see you Wednesday.
See you Wednesday.
300%. But there is kind of a workaround. So you have students who have accommodations where you just want to print a test to give all your students in class one day. There is a way that you can get those tests out of Blackboard and make a hard copy or paper copy that you can get to your student. So I wanted to actually do that workaround in case you ever find yourself in that situation. So right now, I want to actually go into the area where you have to deploy that test. And you're going to take it as a student. So if you haven't deployed it yet, it's still in your survey test school area. You got to deploy it. It has to be in the lesson area, content area. So I'm in our training course actually, where we have a final exam. And I'm going to show you how I would get to this one. So the first thing I have to do is I have to check the setting of it. So I'm going to go to my drop down menu, edit the test options. And I'm going to go all the way to the bottom. And I'm going to look at what happens after the submission, how those results are displayed. And chances are, you don't have it set up this way. So you're going to, for one time only, change the settings, and you can come back in here and revert back to what you have. You do not necessarily want to know how many points each questions are worth when they're taking the test. So you're going to uncheck your traditional setting, and all you want to check is the answers. So after you pretend to take this out of students, all you want to show is all the answers.